So again, good afternoon, class. I hope you all are doing fine, and as we are making progress every week or twice a week. Now we have last Tuesday we covered PCR, and now we are going to learn about DNA sequencing. So DNA sequencing essentially is a modification of PCR, but it is very different from PCR. So first, let's look at the similarities between the DNA sequencing and the PCR. So, so let's say similarities. So DNA sequencing also needs building blocks like ATP, GTP, CTP, and TTP. They also need a DNA polymerase. Okay. So these are the two things which are mainly common. Now, what are the differences? The difference is very less, but very very significant. So DNA sequencing, one of the reagent in DNA sequencing is di-D nucleotides. Now, whenever we write DDNTPs, this N, right? This means that it covers adenine, thymine, cytosine, and guanine. So all together, it's like cocktail of all the things. So this is what is needed in the DNA sequencing. And again, I mentioned to you the role of DDNTPs. DDNTPs are needed for chain termination. Now the question comes, why do you need chain termination? So we need chain termination. I'll show you the figures later. Is because to generate truncated fragments of target DNA that needs to be sequenced. Okay? I'll come back to that later. Why we need truncated? So I'm going reason by reason. Why do we need truncated? We need truncated because these can be resolved on the basis of size. Why do we want to do it on the basis of size? Because it will help to read the sequence of interest. It is the gene that we want to sequence of interest. So there is a logic for everything. So that's one of the major things. The second thing that very critical is that, so in PCR, let's say if this is the target gene, then we need to we use primers for both the strands. Primers are the short oligonucleotide single-stranded DNA that defines the extent of the gene that needs to be amplified. Right? I mentioned to you that if we want to amplify insulin gene, then we don't need to amplify hemoglobin gene or any other genes. So these primers, they define the target genes, both. So that's PCR. In DNA sequencing, you're not doing amplification, right? You simply want to uh, you simply want to read every single nucleotide base that compose this strand, right? And pay attention, if you know the sequence of one of the strand, then of course you can find out the sequence of the pairing strand, because if there is A on one strand, there will be T on other strand, or vice versa. So in PCR, you need to amplify, we need to amplify both the strands. But in DNA sequencing, we don't need both. We need only one strand, right? And so we can get away with only one primer. We don't need two primers for sequencing. So what are the major differences? Number one is that one primer. Number two, we need didenucleotides. And then in DNA sequencing, it has evolved with time. So you might need to do PCR based, like, so you need Taq polymerase also for, but these are the two major differences between PCR and DNA sequencing. Okay, so let's go back to the slide. And why do we need sequencing for? Because we want to know the content of nucleotide bases in the gene, right? Now, when you use the word gene, generally it could mean that segment of DNA that codes for protein, because our DNA, human DNA, is very big. And if you look at the chromosome, right? If you look at our chromosome, then what will you see? You will see that there is a centromere, and this centromere has is attached to, in a duplicated chromosome. There's a pair of chromatids. Right? Within this, there is DNA. Right? Chromosome is a complex of protein and DNA. Right? So you want to understand each and every nucleotide that is present in a particular chromosome. Why? Number one, there are many diseases associated with genes and its function. A mutation can have a dramatic effect on the life of an individual. So the only way we can know what is the arrangement of nucleotide is through sequencing. And once we know that what is the sequence, then you can design the therapeutics around that mutation, right? So, but what is the issue with genome editing, right? Genome editing issue with one of the major issues with is the human cells. For example, this is the cell membrane, and within the cell membrane there is a nucleus. Within the cell membrane there is a nucleus. Now, and within the nucleus there is chromosome, and within the chromosome there are DNAs. think about it, when you want to design any therapy, first, it has to go through the cell membrane, right? Then after it goes through the cell membrane, it has to go to the nucleus. Nucleus has spores. It could be, selective, right? And then once it goes into the nucleus, it has to correct the DNA, the faulty DNA. So it's a multi-step process, right? Now think about it, that instead of changing the DNA, it is possible. But why not, instead of changing the DNA, why not to develop an approach? Because what does DNA do? DNA do? A DNA transcribes into mRNA, and mRNA then goes out of the nucleus to the cytoplasm on the ribosome to translate. So why not to, instead of editing the gene which is in the chromosome, which is very hard to reach, why not to work with mRNA? Right? Because for mRNA, just after getting transcribed, it comes to the cytoplasm. So why not to work with mRNA? Why to have this cumbersome process, two-step? So when people realize this, that, okay, let's work with mRNA, right? So then technology developed for mRNA. And it developed so fast, quickly, that this COVID-19 based vaccines are mRNA based because it is easy. And also mRNA can have less immune insult, right? Anything that goes into our body is foreign. But when we get vaccine in our muscle, mRNA doesn't elicit any bad immune response because of its innate structure, and it can be modified. Also, let's say you can ask me, Professor, why not DNA then? Why don't we have a DNA-based vaccine? Again, that DNA-based vaccine has to cross cell membrane, nuclear membrane, and then chromosome. And there are chances that DNA vaccine can integrate into the DNA. So if a foreign DNA integrates into our chromosomal DNA, then it can cause mutation, because DNA can do recombination 1 in a million times. And it happened that I believe in Europe, kids were given vaccines for measles or something using a DNA-based vaccine, lentivirus-based vaccine, and these lentivirus could integrate at a very wrong places in the chromosome of these kids, and many of these kids developed cancer, blood cancer. So when this incident happened in Europe, scientists were thinking of alternative strategy, because DNA is too risky. You cannot control, you can make tools, you cannot control how they will act inside the cells, very difficult to depict. Maybe artificial intelligence could depict that, but it is same with cancer treatment also. It's hypothetically, theoretically, every chemotherapy agent should work or could work, but it doesn't happen because cells can be very complicated. So unless we know the problem, the DNA sequencing or sequence of a virus or a new strain, how can we? So sequence is knowing sequence of an organism, including human being or virus, is good, is beneficial for not only knowledge, but human health and disease. So sequencing is very important. So many organisms, right? So I don't know. I always struggle pronouncing this name, Neanderthals. People are trying to get some fossils in various parts of the Europe, and with those fossils, people are trying to make a conclusion that they had a very organized and they were very sophisticated at that point of time with the type of life they were leading. So going into evolution is very, very, has come into forefront, that people want to know how things evolved on this planet, how life evolved and in which direction it went. what were lost and what is going forward, right? Evolution, and that evolution can answer many questions. So Human Genome Project is directly related to sequencing. And then I mentioned to you a genetic disorder. Unless we know which gene is affected, which sequence is affected, we cannot design the therapeutic modalities. Then for cloning, right? If there's a gene of interest that can be used for research, then cloning is important. So, and then to confirm various data like PCR data. Because when PCR is happening and it is generating millions of copies in 1 hour, there has a chance, there's potential chance to incorporate errors. Because remember, you can say that PCR is a mechanized replication of DNA occurring inside a machine, but many enzymes that are present in our cell during replication, I repeat, many enzymes that are present during replication inside the cell is not there in the PCR tube, and that leads to incorporation of various mutations. All right. So the PCR data has to be confirmed. For example, Taq DNA polymerase may not have same kind of proofreading or ability to fix the editing in a tube as compared to cellular environment, right? So, so you have to look at this data, and this was the techniques which was in 80s, 90s, and so if I had to sequence HIV genome, I had to use this kind of procedure. And what you see here, this is a picture of an X-ray film. And each of these bands, right? Each of these bands corresponds to one nucleotide, right? And so you can resolve nucleotides in each lane. So this could be a gel, and there could be 4 different wells, and each well will have bands, and you can read the sequence. Now, the problem with this was we used to use radioactive material, and every radioactive material loses its potency, its intensity day by day. So if you are using a radioactive such as phosphorus 32, then it loses its in 14 days, 15 days, and so the bands which are very sharp, right, crisp, they can diffuse. So that was the limitations. And second, you're exposing yourself to the radioactivity, right? Radioactivity can damage the DNA. So these were the conditions in which we were working. Number one, radioactivity, which could have a hazardous effect. Half-life is less, and then so you had to be, not only design experiment, you had to be good X-ray technician, because you have to develop those X-ray and then read, and really you had to break the eye, look very sharply with a lot of focus. So you're not only scientist, not only molecular biologist, but you have to be a good X-ray technician to develop this. So these were the conditions in which we did sequencing of the first HIV strain in India. But slowly, what happened that alternative came to be developed. And right, I think in late 1990s, what happened that people stopped using radioactivity, and then came the fluorophores. Fluorophores are those dye that can emit light when a high energy beam falls on it. So for example, there are 4 bases ATGC, so there can be 4 different dyes, right? And when the light laser falls on these fluorophores, then they can emit different light. For example, A can appear green, G can appear black, T can appear red, and C could appear blue because that's the color of the dye. And this at one time, this can measure around, this one says, almost 350, 370 base pairs, but it could, now it can go till 1,000, 2,000 base pairs, and it can be done in few hours, right? So sequencing technology changed so much. And remember one thing, that technology is going to keep changing, right? So this is where we are now, right? We were thinking about going online maybe next 5 years, 6 years. We were preparing, we didn't know the consequence of online teaching, but suddenly this pandemic happened, and then we are all in a different pace of life. And think about it, how much information has been consumed in past 2 years regarding movement of virus and health conditions. So technology will continue to change and more faster. What was supposed to happen after 10 years, 5 years with regards to online teaching has happened now. So as they say that necessity is the mother of invention. So this developed, and but despite technology can go into leaps and bounds, what is important for you is to have your basic concepts on your fingertips. Because if your base is strong, you can deal with technology. It might take day, 2, 3 to get over the technology. So technology is just like a medium, right? But I still need to know the structure of DNA, right? I still need to know. I can take out the YouTube and I can show, okay, this is the DNA. I may not teach, just show the YouTube video, but that would reflect, the sense of academics. So technology will, that's why, I emphasize right from the first day of the syllabus, discussion on syllabus, that pay attention on fundamental concept, because that's the only constant when the technology is changing. So DNA sequencing moved from radioactivity, from X-ray usage to fluorophores, and the speed of sequencing, it just increased dramatically, speed and time both, and price also went down. So let's go deep into the sequencing. So sequencing is nothing but what we do. We put couple of reagents inside the tube, right, and let the reaction happen inside the tube, right? Let the reaction happen inside the tube. So, but still, there are specific things for each techniques, right? So, what is that key feature of sequencing? So, the key feature of sequencing is that we need 4 different tubes, 4 different, you can say, sequencing tube or PCR tube, and each of this tube has to be labeled with specific dd-GTP, dd-ATP. So in this tube, everything will be the same. Only thing that is different here is dd-GTP. In this tube also, everything is same like orange tube, but this tube will only contain ATP. Similarly, CTP and TTP. Now, why this is happening? I'll come back to that later, so hold on to that. So of course, we need a template, which is a gene of interest that we need to sequence. It could be spike protein, it could be HIV envelope protein. And talking about HIV, you must have read that recently, it was for the first time that somebody turned HIV negative. That's happened because of stem cell transplant. A stem cell transplant was done on an HIV positive patients. And this stem cell had that gene which was resistant for HIV infection. So the patient got this HIV resistant stem cells into his or her body. And then slowly this stem cells grew and grew and grew. And then the patient became resistant to HIV. So that was sort of a first case that has happened just recently. I read this a couple of days back. So more, things are going to happen in next just 1 or 2 years. So we need a gene of interest, and then for sequencing, we need only one primer, because sequencing of one DNA sequence is good enough. And then you have DNA polymerase. This is also a polymerase-based technique, which means that we need to synthesize new strand of DNA based upon the template. You need the regular DNTPs. Again, I said that N stands for all the 4 nucleotides. And then we need DNTPs, DDNTPs. But DDNTPs do not go on every tube. For example, this tube will have, all this tube will have template strand, all this tube will have primer, all these will have DNA polymerase, all these will have DNTPs, but DDGTP only in one, DDATP only in one, DDCTP only in one, DDTTP only in one. Okay. So then of course, when you're putting reagents, you need to put buffers. Buffer, what is buffer? Buffer is a solution that in which the reaction can happen. For example, if certain solution which needs pH of 7, if you put a solution that has pH of 8, it will not work. So buffer is a medium. It is mainly composed of water, but certain salts that will help the enzyme to mediate its function. Okay. So of course, everything has to be, the sequencing now has to be read on computer. Okay. So these are the steps of sequencing. You need template in all the 4 tubes. As I mentioned, you need buffers. You need to add DNTPs, but there will be 4 different tubes, and each tube will have a different DNTPs. And you need enzyme, and you need PCR machine, and this could run for few hours, and then you need a sequence analyzer. So now let's go into the what is exactly happening inside the PCR tube. So before I explain, I go to this figure. So we are same here, right? We are till this point we are with each other on same platform. Why I say that? This is the target DNA. There is one primer that can sequence this DNA, this strand. There are 4 tubes: DDG, DDA, DDT, and DDC. And of course, all of them have polymerase, all of them have primer, all of them have targets. So what is happening now? So let's go to the whiteboard. So so let's take a hypothetical sequence. Right? A, T, C, G, A, T, G, C, A, T, C, G, C, G, T. Okay. So for sake, we are going to only sequence one strand of DNA, right? So that's, and from there we can find out what's the complementary strand, the second strand. So we have to, the goal of this experiment is to read from one end, right? So the way it works here is the chain termination. So here, this chain will terminate at A, chain will terminate at T, chain will terminate at G, A, T, G, right? So at every nucleotide, the chain terminates. Chain terminates mean the polymerization of the complementary DNA stops. Why does it stop? it stops because in each tube, there are 4 tubes. In tube number 1, you have DD-GTP. In tube number 2, you have DD-ATP. In tube number 3, you have DD-CTP. In tube number 4, you have DD-GTP. So you have this 4, right? Now, let's look at this way. So let's say in tube number 1, you have GTP. In tube number 1, you have GTP. So this GTP, DD-GTP, will go and bind to this, right? Then again, the same will bind to this. Same will bind to this here, because guanine will complementarize to cytosine. Now, because of this, right, what will happen? There will be a fragment this long, then there will be a fragment, this long, and there will be a fragment this long. Right? Now, where will DD-ATP stop? So DD-ATP will stop wherever there is T. So DD-ATP will stop at T, because DD-ATP will bind to thymine. This will bind here. This will bind here. And this will give rise to this chain, then this chain, and then this chain. So ultimately what happens, right, with all these 4 tubes, in all these 4 tubes, you will get something like this. You will get fragments like this. Now, if you take some total fragments, you'll get something like this. Then you resolve this fragment, let's say in the classical situation, on a gel. So gel is a matrix where you use electric current to resolve things, because it is DNA. It will move from negative end to positive end. While the current is moving from negative to positive, these fragments, all these fragments will resolve. How will it resolve? The higher one on the top, right? Now, why did I draw this picture? Because in this gel, there are 4 wells. In this gel, there are only 4 wells. Well number 1, well number 2, well number 3, and well number 4. Sorry for my drawing. So well number 1 will have all the strands truncated by incorporation of DD-GTP. This one for DD-ATP. This one third for DD-CTP. Right? So I mentioned to you that there are 4 wells. 1, 2, 3, and 4. 4 wells. There are 4 wells, and each one will have a tube will have sample on them from different tubes, and then you can resolve. So let's go back to the slide. So what is shown here is that this is the target gene, and this is the single strand on which the primer is there. So read the sequence of this. You can read the sequence for sake of learning, right? Of course, in an experiment, you don't know what's the sequence of the target gene. But let's say this is a target gene with the sequence GAC. TAC, GGA, and CGT. You have a primer. A primer is a single oligonucleotide, single strand oligonucleotide that is complementary to the target gene. So you have the primer as sequence as CTG. Now, if you can say with me, if you can, just tell me what will come here, what will stop the reaction here. Here the reaction will be stopped by DD-ATP because it is T here. Here DD-TTP, here DD-GDP, here DD-CTP, again here DD-CTP, again here DD-ATP, DD-GTP, DD-CTP. All these things will ultimately generate a fragment of this way, the way I told you. So sequencing reaction will go on simultaneously in 4 different tubes. These tubes are different on the basis of DD nucleotides. So ultimately, when you put everything together, you get a cumulative band of spectrum of these bands. And when you resolve this on the gel, what is a gel? Gel is a matrix on which you can resolve the DNA on the basis of size or any molecule. The higher size is on the top because it moves slowly. The lower size moves fast, right? So it is faster. Here you can read the sequence, right? So you can read the sequence as this is A, T, G, C, C, T, G, C, A. And you can see that the smaller size A, this corresponds to DDA, this corresponds to DDT, right? This corresponds to DDG, right? So A, T, G, C, right? So see here, this sequence here, because you can look at the opposite end. A, T, G, C, C, T, and this is how it has been resolved. So you can see the sequence A, T, G, C, C, T, G, C. Right? So this is the way the sequencing is done inside the tube. And now it is very fast. You can get 100,000, multiple thousand in few days. Thousands you can get, I believe, in one day, or multiple of thousands. So this is a very, again, chemistry-based technique. And the whole story revolves around the DD NTPs. Both the carbon atom on the ribose sugar, they don't have oxygen. And because they don't have oxygen means hydroxyl group taken together. And this hydroxyl group is very important for chain elongation, for the formation of phosphodiester bond. If the phosphodiester bond is not there, right, then how will the DNA strand grow? Right? So DNA strand has to grow in this direction. And why I'm emphasizing this? Because now we are going towards artificial gene synthesis. Right? So do you have any question, class? So if there are no question, then I'm going into gene synthesis. Now, what we used to call gene synthesis has now changed, because now scientists are synthesizing mRNA very fast. And I told you what is the advantage of synthesizing RNA. or mRNA, because there is less steps involved in mRNA delivery as compared to DNA. So scientists are trying to master the the synthesis of RNA or mRNA. And I've been reading a lot, these days about RNA strategy and synthesis and what are the complications, that I'm thinking to develop a course just on RNA, because RNA is the world now. Of course, that doesn't take away the significance of DNA, but RNA is I think showing the way for future therapy, research, and many other things. So while you want to synthesize DNA, same can be applied for the synthesis of mRNA. But as I mentioned to you, mRNA we will discuss in a separate class. Today we will or maybe next class, and I will finish this chapter with gene synthesis. And so either I dedicate one class for mRNA, or I can teach as we go a little bit information every time. So gene therapy is important. Synthesis of primer is important. All this can happen if gene is synthesized, right? How can we generate primer without synthesis? So what is the difference between the natural replication? Natural replication occurs in 5 to 3 prime, whereas chemical synthesis occurs totally opposite direction. So you can think the ability of nature, how it makes things, so fast. So a typical DNA, a cell, depending upon the type of cells, a cell could duplicate in 24 hours or 12 hours. And then inside our cells, inside our body, the replication is in totally different direction as compared to what is happening in the tube. So you can also look at it in a different way, that in the nucleus, which is such a small, nucleus, which is such a small organelle, that has ability to, that has an environment the chemical environment to let the chromosomes duplicate, replicate, but that cannot be replicated in a chemistry lab. And that's speaks to the chemical aspect of nature. And plants, they are considered to be the first biochemist on this planet. So whenever plant developed, and plant had the capacity as a primary producer to convert solar energy into sugar in chlorophyll, that was the first chemist or biochemist on the planet Earth. Nobody, I mean, nothing is as big as plants in terms of mediating the chemical pathways, right? So so what are the challenges of synthesis in a lab, right? So just let me briefly talk to you about it. Then you will get an idea why things take time in artificial condition as compared to, or let's say why it is so difficult to duplicate what is happening in nature in a lab. So let's say this is a compound. Let's don't name anyone. Let's name it as compound 1. Right? Now, this compound 1 has various phases in which the chemical reaction can occur. Here, it can occur here, it can occur here, it can occur here, occur here. But inside the cells or biologically, the reaction is only occurring here. The reaction is only occurring here at this position. Now, this could be possible inside the cell, but how it is possible inside the tube? Right? In a glass tube or in a plastic tube, how it is possible? Right? So in order to do that, what scientists do, that they try to block the unwanted site. So they will block this side, they will block this side, they'll block this side, they'll block this side. When they block, or let's say when you engineer a molecule that has blocked all these sides, right? Then only it is possible that reaction will occur here, right? Otherwise, there are equal chances that reaction can happen in all these 5-6 places, and that will reduce the efficiency and increase the impurity in your chemical mixture. So how will you? So ultimately you want this. So this will be 20% of 100% what is being made. You don't want that. You want more efficient. So you get these compounds. So we get a artificially synthesized nucleotide that is called as phosphoramidite. You don't need to go into the chemistry here in this chapter or in this class. You need to understand concept. Of course, there will be a question, but the question will be of very less. You don't have to draw chemical structure. You don't have to know the chemistry, oxidation, reduction. You just have to have concept that what happens at what step, and that's good enough for this class. So for any reaction, there has to be a starting point, right? There has to be a starting point. So, and when things are happening inside the tube, right? When we finish the reaction of gene synthesis, then how are you going to take out the gene? Gene could be, into water, into mixed with impurities. And how do you extract the gene that has been newly synthesized? So one way to do is to have that target gene or target chemical or starting point tagged to a bead. This has a weight, this has a density. So if DNA has been synthesized on this bead and you centrifuge this, centrifuge is a machine where you apply, it runs and then it throws thing inside centrifugal, and then centrifuge machine, and then you can, anything that is tagged, so this is heavy, so this is settled down in the bottom of the tube. And then whatever is there with this bead is your new DNA, hopefully. But if this is the bead, you cannot start the reaction right away. You need to have a spacer. So this is a bead, which is made up of control poured glass, silicon, and they can settle down under the force of centrifuge. And then there's a spacer. And after this spacer, there is a first nucleotide. Now remember, the first nucleotide in artificial synthesis is actually your last nucleotide, because in natural condition, the synthesis will occur from 5' to 3'. Here it is 3 to 5'. So this is your beads, this is the spacer, because if you do just reaction very close to beads, it may not work. So all these chain, the spacer has been optimized. When we talk about mRNA structure, you'll also know that the start codon is just not next to promoter. There is a spacer. There is an untranslated region. Perhaps that helps various proteins to come and to start or helps the ribosome to sit on the start codon, and then it is just not adjacent to the promoter. So that RNA polymerase may not compete with ribosome. So various kinds of hypothesis and speculation, but this has been optimized, that this distance is optimal for a good synthesis. And the precursor, right? I said that we cannot put a nucleotide here, because if you put a nucleotide, true nucleotide, the synthesis can happen in many directions. We don't know. So we need to take it in a very, very controlled way. So to do that in a controlled way, we need a molecule called as phosphoramidite. So this is the molecule. Now look very closely, it's very interesting. When you look closely, you will understand that there's a nitrogenous base, there's a ribose sugar, but instead of phosphate, right, instead of phosphate, you get such a complex structure, chemical moiety, diisopropyl amine group. You don't need to know the structure. So you get all this. So why all these moieties are here instead of just a phosphate molecule, right, of a nucleotide? The reason these are there because the phosphate, phosphate is what, PO4. So one of the oxygen is being covalently attached to the third carbon atom, but rest three oxygens have been blocked. They have been blocked because only that oxygen goes and forms phosphodiester bond with the fifth carbon atom that is most needed. This is blocked, right? So as I mentioned to you in my white slide, right? So this is the starting base for this is phosphoramidite. So we don't need this DMT, we don't need these blocked agents, then only we can make the gene, right? So that's the process of synthesis, to remove the blocked regions and then facilitate the formation of covalent bond in the right direction with the right oxygen atom in the phosphorus to make a good phosphodiester bond. Okay. So first step is to remove the DMT. DMT stands for, DMT I wrote here, it stands for dimethoxytrityl. So DMT, so this DMT, this DMT has to be removed. Now it's not like restriction enzyme that you cut this bond. No, you have to treat them with chemical. So in the first step is to remove the DMT so that this oxygen becomes available for the formation of phosphodiester bond for the incoming base number 2. So this is done with the help of a chemical TCA, that is called as trichloroacetic acid. And a simple step can remove the D. So this is the first nucleotide with OH free. This is the fifth carbon atom and the OH hydroxyl is free. So the next base has to come and they have to form a phosphodiester bond. Right? So what happens to the next base? So, right? So this is the pre-existing, the CpG bead, spacer, and the first base that has undergone removal of DMT. The next phosphoramidite that comes as base 2. So this has to be activated. And this is activated chemically. And this activation leads to the protonation of this nitrogen. Now look at the position of this nitrogen. This nitrogen is attached to this phosphorus. So let's look at this phosphorus. Right? We want to create a phosphodiester bond, right? So let's look at this phosphorus carefully. Right? If you look at this phosphorus carefully, what do you see? One of them is attached to the oxygen that goes to the third carbon atom. One of the oxygen is attached to this methyl group, and this is attached to a big moiety here. So the first is to activate this nitrogen, and this nitrogen gets protonated, right? Loss, because positive charge, in simple words. Now, next, what happens here? So this gets protonated, and after some, I'll tell later that this nitrogen and whole moiety is removed, and this phosphorus, so this becomes protonated, positive charge, less electron, This hydroxyl is reactive group. This attacks this here, nucleophilic attack from this hydroxyl group. And when the hydroxyl group attacks this protonated nitrogen, coupling happens. And this coupling leads to the formation of addition of first base, or the formation of the first phosphodiester bond. But still, here you have to remove this methyl group and you have to add one oxygen. It has to undergo oxidation. So first reaction is detritylation. Second is activation to create a protonated nitrogen. And then there is a coupling. Coupling is facilitated by nucleophilic attack by this hydroxyl group. And then, now while this reaction is happening, there are some unmodified nucleotides, and they can start the reaction on their own because they are all part of the tube, single tube. So they have to be capped. So capping of unused or unutilized bases prevents interference by these moieties, because chain is growing, right? It happens in cycle. But chains could get disrupted, and disrupted chains have to be stopped because they'll start growing at a different time. So this stopping is done by capping. Now here is one of the most critical steps. So here, this oxidation happens, and then oxidation leads to the formation of third oxygen. So first oxygen here, second oxygen here, third oxygen forming the phosphodiester bond, and fourth is blocked with methyl group. So this is the basis for, and I have written here, I don't want to read, I don't like reading slides, but read these specific details about detritylation, coupling, and then capping, and oxidation for generation of phosphodiester bond. So to sum up, in this chapter, we looked at the PCR. I made PCR as a model to understand synthesis. Then we use DNA sequencing to understand how gene is sequenced by dideoxynucleotides through chain termination, and then sequencing. So all these three processes are very interrelated. So with this, I finish this chapter, and I have put the recording for the first part, and now I will upload the recording for the second part. So I will stop sharing my slide, and I look forward to receiving your or listening to your questions so that I can address them. Questions, class? So if there are no questions, then can we end this class? No response.
Night, everyone.
Okay.
Professor, do you have my
Professor?
name?
Yes.
You have my name?
I'll mark you present. Tatiana Ross. Anybody else?
Do you have my name, Professor?
What is your name? Just tell me, because people are speaking it simultaneously. So Tatiana Ross and?
Kiara Donald.
Kiara Donald, yes, I got you.
Thank you, Professor.
Okay. Tatiana Ross, anybody else?
Yael Nakhshumov, do you have me?
Who?
Yael Nakhshumov?
Yael Nakshima, yes.
Yes.
I have.
Professor, did you get my email? I tried to submit the assignment via Blackboard, the first one with the questions, and I wasn't able to on Blackboard, so I sent it by email.
No, I never got your email, and That's why I don't tell people, I don't encourage people to communicate, send assignments through email, but I saw your assignment on the Blackboard.
I'm asking in regards to the assignment with the questions, not the one, the introduction one.
No, I didn't check the second one. I'm talking about, because today is the last date for submitting the verification of enrollment attendance. So I focused on that. You want me to look there?
Yes, this one I submitted. This one I know. I'm just
Okay.
asking about the one with the questions about the first chapter.
Okay, let me just check, and I'll get just be patient.
Sure.
Remind me your name.
Yael Nakhshomov, Y-A-E-L, and the
Lakshma?
last name is
No, I
N-A-K.
don't. I don't see here, and I didn't receive your email.
Okay, so I'll send it again today.
No, upload that. Don't send me an email. Email is not guaranteed. That's why I discourage
Okay.
students to. So
Okay, so I'll
you have
put it.
told me, so I believe you, but
Mm-hmm.
try to fix your Blackboard, okay?
Okay, thank you.
Yeah, email is very, very more unreliable than Blackboard. Blackboard is very solid. Sometimes things happen, but mostly it is very reliable.
Okay, so I'll try to upload it again.
Yeah, try that. But you told me,
Okay, thank you.
Okay. Any other
Okay, thank you,
question?
Professor.
Professor, do you have my name? Aisha?
Aisha?
A-I-S-H-A.
Let me just check.
Orange.
Yeah, I got you.
Thank you.
Any other questions?
No question. Have a good night, Professor.
Good night.